Welcome to this video on semi-conservative mode of DNA replication, or you can say molecular basis of genetic inheritance. After DNA was proven to be the genetic material, the next pertinent question was how does DNA transmit information from one generation to another? There were three hypotheses prevailing at that time, including dispersive, conservative, and semi-conservative mode of replication. Watson and Crick proposed that DNA replicates in a semi-conservative manner, meaning that each daughter double-strand DNA molecule consists of one original parental or old DNA and one newly synthesized strand of DNA. This hypothesis was experimentally confirmed by Meselson and Stahl in 1958. In this video, I will explain the Meselson and Stahl experiment, their finding, and the conclusion, which rejected other models and confirmed that DNA replicates in a semi-conservative manner. Because of its elegance, simplicity, and conclusiveness, this is often referred to as the most beautiful experiment in biology. Before coming to the experiment of Meselson and Stahl, I would like to briefly describe these modes of replication. So let us begin with conservative mode of replication. According to this, the entire old double-stranded DNA is conserved completely and a completely new strand of double-strand DNA is made. So this is conservative. According to dispersive mode of replication, old and new DNA are mixed in bits and pieces. Therefore, both strands of each DNA are a patchwork of old, shown here in red, and the new, shown in green. This is dispersive mode of replication. According to semi-conservative mode of replication, 50% of DNA is conserved. It means each daughter double-strand DNA has one original and one newly synthesized strand of DNA. This is semi-conservative mode of replication, and this was addressed by Meselson and Stahl. The conservative and dispersive replication could not be explained by a convincing mechanism. However, semi-conservative mode of replication was explained by Watson and Crick. According to them, the parental DNA strands separate and they serve as template to synthesize new strand of DNA. Therefore, daughter double-stranded DNA has an old and a new strand of DNA. Before describing his experiment in detail, let us review the components of his experiment. The first one is E. coli bacteria. To label its DNA, he grew bacteria in the growth medium containing N15 heavy isotopes. In this condition, the DNA will be labeled with heavy isotopes, and therefore this will be heavier DNA or dense DNA. In a separate batch, he grew bacteria containing N14 light isotopes of nitrogen in the growth medium to label DNA as lighter DNA, and to separate heavier from the lighter DNA or dense DNA from the lighter DNA, he used cesium chloride solution. When cesium chloride is centrifuged, it forms a gradient, and biomolecules or these DNA can be separated easily based on their density. So N14 will stay up and 15, which is heavier, will stay down toward the bottom of the tube. Regarding the experiment, they grew bacteria in presence of N15 heavy isotopes of nitrogen for several generations, and they isolated DNA run on centrifugation, cesium chloride gradient centrifugation. The one band was isolated corresponding to heavy DNA and 15 DNA toward the bottom of the tube. In a separate experiment, they grew bacteria in presence of only lighter isotopes of nitrogen and 14. DNA was isolated and run on cesium chloride gradient. One band corresponding to 49 DNA or lighter DNA was obtained toward the top of the tube. This experiment basically confirmed that these heavy and lighter DNA can be separated nicely on cesium chloride gradient. It was also important to it also important serve as a control to compare density of DNA from experimental condition. Now the bacteria which were grown for several generations in presence of heavy isotopes N15 were transferred to new growth medium containing only N14 or lighter source of nitrogen, and aliquots of culture were taken out at first generation or 30 minute, second generation or 60 minute, third generation of 90 minute. When DNA was isolated from first generation bacteria, only one band corresponding to the medium density was obtained, it rejecting the conservative mode of replication. However, both possibilities are there. Both semi-conservative and dispersive replication can produce this medium density band. Therefore, to find out which one is producing this band, DNA was isolated from second generation, run on centrifugation, the cesium chloride gradient centrifugation. They obtained two bands in this case, one corresponding to medium density and the other one corresponding to lighter DNA. This ruled out the dispersive mode of replication, because in dispersive mode of replication only one band corresponding to medium density was expected. However, because both bands are appearing here, semi-conservative mode of replication is most likely. Therefore, to further confirm that, they isolated DNA from third generation bacteria, run on cesium chloride gradient, and they saw two bands appearing again, the medium density DNA and the lighter density DNA. However, the amount of the lighter DNA was roughly about threefold of the medium density DNA. This suggested the semi-conservative mode of replication and rejected the other two, including dispersive and conservative mode of replication. So in conclusion, these findings supported semi-conservative mode of replication and rejected completely the dispersive and conservative mode of replication. So thank you very much for watching this video. I request you to like it, share it and subscribe it. Until next time, you take care. Bye bye.